Hello, students. All right, today we are going to continue with topic 10.2, and we are going to be looking at linked genes. So let's put on our thinking caps. All right, so linked genes mean pretty much what the essential understanding here is saying. Gene loci are said to be linked if they are on the same chromosome. So unlike what Mendel studied, he pretty much got lucky and he saw gene loci that were on separate chromosomes. That's not what linked genes are. They happen to be on the same chromosome. So this is something very important that we have to remember. So a linkage group is a group of genes whose loci, remember loci is the position, are on the same chromosome. Hence, they cannot independently assort. So they cannot do what we've been doing in Mendelian dihybrid crosses. So linked genes will tend to be inherited together. So they don't follow normal Mendelian inheritance for a dihybrid cross. Instead, the phenotypic ratio will be more closely aligned, looking more like a monohybrid cross, as the two genes are inherited as a single unit. Now, linked genes may become separated via recombination due to crossing over, but that all depends on the location of where the linked genes are. The further apart they are on the chromosome, the greater chance for recombination to occur. All right, so let's look at how linked genes are represented. So anytime you want to show that via a notation, you show it like this. So I don't usually show two lines here, but you can. The two lines represent the two separate chromosomes. I usually just do one line like this, but you can draw the two lines. I guess that's a more correct way of showing that the two genes are linked, but happen to be on separate chromosomes. Okay, so let's look at what Mendel saw. So Mendel looked at unlinked genes, and he studied a dihybrid pattern of potential traits in pea plants. So what Mendel saw was he saw two different traits. Let's call them A and B. Now, he didn't know that these two traits happened to have genes or loci or alleles, let's call them that were found in separate chromosomes. So here you see the two alleles for this particular trait. They happen to be on separate chromosomes. And here you see the two alleles for this trait or that gene that happen to be in two separate chromosomes. So then we learned when we were looking at Mendels experiments that we need to FOIL to independently assort. So the law of independent assortment, which was Mendel's second law, took an effect here, right? So you could potentially assort one allele, the first alleles together to give you this gamete, or you could assort these alleles, the outer ones, to give you this gamete, or the inner ones to give you this gamete, or the last ones to give you this gamete. So in Mendel's dihybrid pattern, you saw four potential gametes, which if then you took this heterozygous genotype and you crossed it with another heterozygous genotype, you would always get the 9 to 3 to 3 to 1 ratio, phenotypic ratio, right? So here you see how these four possible gametes are shown to be independently assorting. And if you cross it with another heterozygous individual, the gametes will also independently assort. So then you end up with a 4x4 table in your 9:3 to 3:1 phenotypic ratio. Okay, so again, this is what Mendel saw in his experiments, and he saw genes whose different alleles were found on different chromosomes. Okay, and the law of independent assortment is being represented. All right. So what are linked genes then? How can we show it in a picture here? So this is what a scientist known as Thomas Morgan saw. He saw these linked genes that follow a monohybrid pattern instead of a dihybrid pattern. So it's kind of like how you would do a dihybrid cross, a Mendelian dihybrid cross, when you do it incorrectly. So in this particular case, notice how you have the different alleles, right, for that one gene located in two separate chromosomes. But notice how the alleles are linked. The A goes with the B, or the dominant A goes with the dominant B, and the recessive A goes with the recessive B. So because they are linked, because they are in the same chromosome, they are not going to independently assort. Therefore, they will go together to that gamete. And the same thing here. These two go together to this gamete. So instead of ending up with 4 potential gamete combinations, now you're going to end up with 2 potential gamete combinations, right? The big A, big B, and the little a, little b. Okay? So this is what we refer to as a heterozygous linked combination here. Now, if you were to cross this heterozygous individual whose genes are linked with another heterozygous individual whose genes are also linked, then you would end up with a simple monohybrid cross, a 2x2 Punnett square, where you would just simply put the gametes together like so, these two, right, this one, and then this one and this one from this guy and this guy, okay? And then you would end up with different possibilities, right? In the offspring, you would end up with this type of possibility. That's this guy right here. You could end up with this type of possibility. That's this guy right here. This and this are the same, right? But you could also end up with this type of possibility, like that, okay? All right, so this is what a linked Punnett square looks like, as opposed to an unlinked Punnett square looked like. So again, here you're following Mendel's law of independent assortment, and here you are not following Mendel's law of independent assortment. So Mendel did not even know that there was such a thing as linked genes. So like I said, he got lucky when he studied the genes that he studied because they did independently assort. All right, so let's talk a little bit about Morgan's discovery. So Morgan, Thomas Hunt Morgan, he did some experiments with fruit flies sometime around the 1900s, so early 1900s, like 1909 to 1914. And he found that his results when he crossed fruit flies did not agree with Mendel. And the way that he compared his results with Mendel's results was through a chi-square analysis, right? Where you take your observed result and you compare it with expected results. So he compared what he saw, his observed results, with Mendel's expected results, and he saw that they weren't quite agreeing, right? So first he thought, well, is it that Mendel made a mistake? Are his results anomalous? Was Mendel cherry picking his results? He couldn't quite decipher it, right? But then he realized that there could be something wrong that Mendel maybe did not account for. And remember that when Mendel conducted his experiments, he didn't even know about chromosomes, right? He didn't even know about genes. And when Thomas Hunt Morgan made his discoveries, he saw non-Mendelian ratios in Drosophila, or these fruit flies. And he said, well, something is happening here with these genes. Perhaps they happen to be on the same chromosome. And this is what he called gene linkage, right? When two genes do not independently assort, do not follow Mendel's law of independent assortment, and where you have genes that go together in that same chromosome and are passed on together in the same chromosome. So back in the 9th grade and in SL, you looked at one form of linkage, of gene linkage. You looked at sex linkage, right? And you saw a little bit of Morgan's experiment when you did a crossbreed, let's say between red-eye wild types with wide-eyed mutant flies. And you saw a clear sex bias in the phenotypic distribution. So let's go down here and see if you remember what you studied back in the 9th grade. So remember that in sex-linked genes, only the X chromosome carries the allele. The Y chromosome does not. So what if you're crossing a white-eyed female, right? And let's say that white is dominant over, here we go, red-eyed female, red is dominant. Red is the capital W, sorry about that, yeah. So red is dominant over white. So what if you're crossing a homozygous white-eyed female with a red-eyed male? So if you remember, in a Punnett square like this, you would do, it doesn't matter where you put the male or the female, little w, and then the male here, big W, and then nothing on the Y chromosome. So then when you did this cross, you got something like this. And I like to make big distinction between the big W and the little one so I don't make a mistake. Okay, so a little W here and a little W here. So you end up with 100% of the males come out with wide eyes. And then you end up with 100% of the females come out with red eyes, right? Because red is dominant over white. So notice that here you're not following your typical Mendelian genetics because you don't carry any allele on the Y chromosome. Okay, so this is how he first discovered linkage, but he called it sex linkage, right? And he discovered that in Drosophila. But there's more than sex linkage, right? What about what about gene linkage of other genes that are not necessarily in the sex chromosome, that could potentially be in the autosomes? So Morgan went on to identify a number of traits in fruit flies that did not conform to Mendelian ratios. He saw that certain phenotypic combinations occurred in much lower frequencies than was to be expected. And again, he studied this via a chi-square distribution. So after he ran his experiments for a period of 10 years, I mean, this was not something that he did once, right? He made two key proposals, and these are important here. One, that the alleles for these traits were located on shared chromosomes, so on the same chromosomes, just what we've been saying since the beginning. So if the alleles are on the same chromosomes, they are linked, and they do not independently assort because they happen to be on the same chromosome, unlike what Mendel saw. Now, he also proposed that some linked alleles could be uncoupled and could recombine via crossing over to create alternative phenotypic ratios, but that these phenotypes would be at a much lower frequency. So this is something that we're going to explore when we look at some genetic problems, that the offspring aren't always going to look like the parents. There's going to be some recombinants. But this all depends on where the alleles happen to be on the chromosome. So alleles who happen to be close together on the chromosome, they have a much lower frequency of recombination, whereas alleles who happen to be far in the chromosome, they have a much higher frequency of recombination. And in the next video, we're going to talk about why that happens and why recombination is more likely to happen when there is a greater distance. And we'll see how crossing over happens in alleles that are far away. So Morgan also observed that the amount of crossing over between linked genes differ depending on the combination of traits. So let's look at this picture here. So this led to the idea that crossover frequency, so recombination, may be a product of the distance between two genes on a chromosome. So this is what I just said. Genes with a higher crossover frequency are further apart, right? Higher crossover frequency are far apart. Genes with lower crossover frequency are closer together. Okay. So Morgan used this concept to develop the first gene linkage map that showed the relative positions of genes on a chromosome. And there's actually a unit that's called a centigram, which is named or developed after Morgan and his positions of these genes or alleles. I'm sorry, not a centigram, a centimorgan. Did I call it a centigram? A centimorgan, named after him. This is not an actual SI unit, but it just helps to convey the length of these alleles within a particular chromosome. So here is just an example of gene linkage and recombination frequencies. So here you see what a heterozygous genotype would look like. So wild type, or the term wild type, means the dominant genotype. So it's the one that you see more frequently. So for fruit flies, for Drosophila, if you have long aristae, these are the tiny little antennas, or long legs, or a gray body, or normal wings. Normal wings means that they're like normal size, right? That's what normal means. Or red eyes, then you're the wild type. You're the dominant. You are the quote-unquote normal type of fly. But then you have some mutant recessive flies where they have a short little aristae, they have short legs, and they have a black body. They have vestigial wings. Vestigials means that they're just smaller. And they have brown eyes. So this is what they call the mutant recessive. So you could potentially cross, and this is what Morgan did, a heterozygous wild type versus a homozygous mutant to see what type of offspring you would get. And this is some of the genetic problems that we're going to start observing in the next video, and I'll work out some of them in the next video.